As I've said before, we spend most of our time in micro talking about the prokaryotic cell. We probably have more experience in dealing with eukaryotic cells. So if you've taken a general biology class or anatomy and physiology class, we tend to spend more time talking about eukaryotic cells, which are a little more complicated. So just to refresh our memory a little bit of the similarities and differences of prokaryotic and eukaryotic cells, prokaryotic cells are very simple cells. So they are the oldest cell type. So this is actually the precursor for eukaryotic cells. There's no nucleus. In fact, it basically means pre-nucleus. No fancy organelles like the mitochondria. They do, of course, have DNA, and of course, they're going to have to undergo all of the things that living things do. They have to undergo metabolism. They have to undergo generative properties. They have to maintain homeostasis. Eukaryotic cells are much larger and they're more complex. They do have several things in common. As I said before, they both share characteristics of living things. They have DNA, ribosomes. If you remember learning about ribosomes, that's where proteins are made. So bacteria, they need proteins just like we need proteins. They both have cytoplasm. And of course, they both have a plasma membrane. They're very, very simple cells. So in this chapter, we're going to spend more time talking about the basics of prokaryotic cells. I'll also give you some examples. We'll also talk about some shapes and arrangements of the bacteria and what we're going to expect to see in lab. So bacteria come in a variety of shapes. As I mentioned before, these are very small. So to visualize them underneath the microscope, we need that oil immersion lens. We're going to need the highest magnification that we can get on our compound light microscope. So because of their cell wall, which is a little different, prokaryotic cells have a cell wall that we'll spend some time talking about. Eukaryotic cells, unless we're talking about plants or fungi, they do not have a cell wall. Cell walls are completely different, so bacterial cell walls are different. One of the things that the cell wall does is that it helps to give them their shape. So these are the different shapes of bacteria and the different shapes that we will be seeing in lab. So a very common shape that we see is a circle. And that shape would be a coccyx. If we're saying plural, it would be cocci. So you'll notice that I really like a lot of letter associations. So I think about circle begins with C, cocci begins with C. And you can see that blue cell that looks like a sphere, looks like a circle, that is a coccyx. If we had multiples, we would call it a cocci. The other common shape that we see on the other side of the screen is the bacillus, or bacilli for plural. And that is a rod-shaped bacteria. To me, it kind of looks like a Good and Plenty candy, or almost like a capsule, like if you're taking a Tylenol, or maybe even a Mike and Ike candy. So it's a little more elongated. Some look a little more like sticks. But it is certainly longer than the circle that we see with the cocci. Some that we'll see are sort of an amalgamation of both of those, and this is a coccobacillus. Coccobacillus, to me, kind of look like a little tic-tac, where it's a little oval, but it still has sort of that rounded shape. Those are going to be the most common shapes that we will see this semester in lab. There are some other shapes. And I'll talk a little bit about some examples of bacteria that would fall under these shapes. One is Vibrio. And Vibrio is a comma shape, or almost like an apostrophe shape. Spirillum has a little wave to it. Sometimes in our prepared slides, we're able to actually see that shape. And Spirochaete is a little more like a corkscrew shape. As I said before, the two shapes that we are going to be seeing the most is the cocci and the bacilli. Now, I know that I use very different terms to describe, just so that we kind of have a reference. I'll call it a circle or a sphere. What I want you to do is to get used to using those micro terms. So instead of saying circle, instead of saying rod, make sure that you're saying coccus and bacillus. We have lots of different terms to kind of describe what we're seeing, but in micro, we want to make sure that we're using the correct terminology. So this is an example of a spirochete, and you might actually be familiar with this particular spirochete. An example is leptospirosis, and this is caused by a spirochete. And humans can get this as well as animals. And so if humans or even other animals are in contact with contaminated urine from maybe wild animals like birds or rats or mice, they can actually contract leptospirosis. Now, there is no human vaccine for it. However, if you have pets, your dogs might have actually gotten a vaccine for this. So sometimes we can actually see this disease arise in places where there's a lot of standing water. So if there's a lot of flooding, if let's say that there was a disaster like an earthquake or a hurricane, and now we've got standing water, this is going to be a quick way for a type of bacteria like this to be transmitted. So this is also the reason why if I'm drinking a can of soda or seltzer, I'm always wiping off the top of the can. One of the things that you will hear me talk about is that my mother was a nurse, and that was one of those cases of a little bit of knowledge was probably too much, because she was, I don't want to say paranoid, but certainly aware of a lot of things. And so one of the things that she taught me to do is to wipe off soda cans or even canned goods before you actually open them, because that could be a way that leptospirosis is transmitted. So if you think about these cans in warehouses, they're not the cleanest places. There are going to be mice and rats running around, and they could actually be transmitted. So my mother at an early age taught me to wipe the cans of my soda and canned goods. A few years ago, I remember hearing in the news, and this was right after the earthquake in Puerto Rico. And of course, there was a lot of flooding. There was a lot of devastation. There are a lot of dogs in Puerto Rico that are running around that don't have homes, that are strays. In fact, my one dog was a rescue from Puerto Rico. And at the time, after the earthquake, of course, they were sending help. They were trying to get supplies in. And one of the things that they were doing was that they were rescuing puppies. So they brought the puppies back to New Hampshire, and they actually had the puppies at a local pizza place. And I honestly can't remember the town in New Hampshire, but I do remember hearing this on the news. And the reason I heard about it on the news was that people that had gone to the pizza place and visited with the puppies because they were trying to get them adopted, they were putting an all call out on the news to say that if you had been in contact with these puppies, you may want to get in contact with your doctor, because the puppies had leptospirosis, because of course they were coming from a place where there was standing water and devastation. So people were warned that if they were at that pizza place, if they had come in contact with the dogs, to make sure that they were going to see their doctor. Again, dogs can get the vaccine. In fact, I believe that both of my dogs have actually had the vaccine for that. An example of Vibrio is cholera, and we've probably all heard of cholera, again, in some areas where there is a lot of devastation and standing water. This is how cholera can be transmitted. And of course, symptoms, lots of horrific GI issues. Of course, if you have GI issues, then you risk dehydration, which is not going to be a good thing. So sometimes we often hear about cholera as kind of a secondary after devastations like earthquakes and hurricanes. We will not be seeing any Vibrio cholera in lab. We will also not be seeing any spirochetes in lab. But these are just a couple of examples. Since we are talking about some bacterial diseases, this is not a human health and disease class. So even though we talk about these diseases, it really is a standpoint from recognition, not a lot about the pathology and all of the symptoms and treatments. We won't be getting into that. It's mainly for recognition. So if you're like, yeah, I've heard of Vibrio, you're going to know that, yes, that is a bacteria. We will also be talking about some viral diseases so that you know the difference between, okay, yes, I can recognize that as being a viral disease. I can recognize that as being a bacterial disease. So it really comes from a recognition standpoint. So another thing that I have mentioned before is arrangement. And arrangement happens when the bacteria are dividing. So that cell wall helps to give some protection to the bacteria. It also is the thing that basically helps it form the shape. When bacteria are dividing, and we'll be getting into how they divide in the next chapter, if they get stuck together, this is when we can see different arrangements. So it's not a complete separation. They kind of get stuck together. So some common arrangements that we will be seeing, you will see mono. So some just kind of like to hang out by themselves. They separate very well during cell division. And so they're not going to be stuck 2 by 2 or 4 by 4. So some you will see that are going to be by themselves. And I usually call that mono. Diplo is when we have 2 bacteria stuck together, and we could see diplo with cocci, so we could see 2 circles stuck together. We could also see diplo with bacilli, where we see 2 rods stuck together. If we saw 4, that would be tetrad, and we tend to see that with cocci, not with bacilli, just because the bacilli actually divide end to end. So if we were to actually draw out a bacilli, and I'm going to attempt to draw a bacilli right here, if it were to divide, it's actually going to divide end to end. So we would see diplo. We could actually see a chain of them together, and that would be strepto. Of course, we could also see strepto with cocci as well. But we tend to see fewer arrangements with the bacilli just because they're going to be dividing end to end, and that's how they would get stuck. Whereas if you think about a circle, they could actually be dividing around that, all of the planes of that circle. So like I said, we tend to see more arrangements with cocci than we do with bacilli. So common arrangements that we will see: diplo, strepto, tetrad. Another common arrangement that we will see is staph. And staph is when they're arranged in sort of a grape cluster, and we would only see staph with cocci. And I'll circle the picture where you can actually see staph. So if you look at these individual cells, they're these little tiny circles, and they're stuck together in almost like this grape-like arrangement. And so to me, they kind of look like bunches of grapes or bunches of balloons. If you look at this picture, it almost looks like lace. So where they're kind of gathered together, and then you kind of see the spaces in the middle. We would only ever see staph with cocci. So the most common arrangements that we'll see: mono, diplo, strepto, and staph. We will see one organism that likes to hang out in arrangements of 4, tetrad. If they like to get stuck together in groups of 8, that would be sarcinae. And sarcinae is kind of like a strange sounding word. To be honest, I don't believe that I've ever seen sarcinae. Sarcinae can also be kind of difficult to see because if it is in tetrad, you might kind of mistake that because you might see 4 up front, but there's actually another 4 behind it. It's possible to also see like rosette or a star shape. I'm going to be honest with you. I've never seen star shape underneath a microscope. I've only ever seen it in pictures. So we are going to spend quite a bit of time looking at cells, identifying their shape and arrangement. So here's my little drawing right here, and this kind of shows you the different arrangements that you can see. If you notice that bottom picture, I have a big X through it. We would never see, unless we had a really thick sample, it might kind of mistake it for this arrangement. We would never see bacilli kind of piled high like a stack of pancakes, just because that's not how they divide. So the only way that the bacilli are going to get stuck together is end to end. So we would see mono, we would see diplo, we would see strepto. As I said before, because of the cocci, because of the circles and dividing on those different planes, that's when we tend to see more arrangements. So this is just a little reminder that we are not going to be seeing bacilli in staph. We won't be seeing them stacked up like pancakes. So we'll be talking a little bit more in detail about the bacterial cell wall. As I mentioned, it is completely different makeup than something like a fungi or a plant that has a cell wall. It's completely different. It helps to give it its shape. It also helps to protect it to a certain extent. And one of the things that we'll be doing in lab is doing some stains so that we can differentiate. And again, one of those differential stains where we can differentiate between bacteria based on their cell wall. So we're going to be looking at a little comparison of our bacteria and the different types of cell walls. But first, we'll talk about some of the components that we might see in a cell wall. So first up is peptidoglycan. Another name for peptidoglycan is murine. This is really the most important part of the bacterial cell wall. Eukaryotic cells are not going to have peptidoglycan. This is going to be something that we're only going to see with bacteria, these prokaryotic cells. This is like the fence-like structure. So this is going to be outside of the cell membrane, and it's going to help to keep things out and, of course, keep things in. We're going to be seeing peptidoglycan, although it's in different thickness in both gram-positive and gram-negative organisms. And before in chapter 4, when I was talking about the gram stain, that gram stain, because it's a series of stains, is going to allow us to see if that organism is gram-positive or gram-negative. That's going to tell us about its cell wall. Now, as far as a clinical standpoint, why would this be important? This would be important because if you have a patient that has a gram-positive infection, you want to make sure that you're finding an antibiotic that is going to be targeting those particular gram-positive bacteria. So this is why it's important from a clinical standpoint to do our gram stain. So we're going to see this peptidoglycan in gram-positive, gram-negative, and even acid-fast bacteria that I'll talk a little bit about. Another thing that we would see in gram-positive organisms is teichoic acid. And we only see this with gram-positive organisms, and they believe that perhaps it has some ion transport function. And this also kind of serves as a docking station for certain viruses. And there are certain viruses that only attack bacteria, and we call those bacteriophages. And so this serves as a docking station for it. If that virus can't attach to a cell, then it's not going to be able to replicate. So that's why that attachment point is going to be really important in regards to the virus and its ability to replicate. We can also see an outer membrane. So we would only see an outer membrane in gram-negative. And when I show you the picture of the gram-negative organisms, I kind of describe this as kind of like a peptidoglycan sandwich, where we'll see an inner membrane, a layer of peptidoglycan, and then an outer membrane on the top. We would only see that in gram-negative. Something else that we talk about with the outer membrane is LPS, or lipopolysaccharide. This is actually an endotoxin, so it can be really toxic if it's found in large quantities. And this is actually something that can cause endotoxic shock, which sometimes happens with the elderly. So if we have a massive kill of gram-negative bacteria, as that cell lyses, it's going to be releasing a lot of that endotoxin, and this can actually cause endotoxic shock where the person has a high fever. It causes blood vessel dilation, and their blood pressure can start to plummet. We can also see in gram-negative bacteria this periplasmic space. So this is kind of like that gap between the cell wall and the cell membrane. Remember, bacteria, they don't have a mitochondria. And if you remember, mitochondria is where ATP is made. So they can actually use this space to undergo some cell metabolism with some of those electron transporters. I'm a visual person, honestly, so I think it's a little easier to see the makeup of the cell wall by looking at pictures. So this is just a little summary page of what I talked about before. So gram-positive bacteria. And one of the things that you will notice is that I like a lot of letter associations. So if we think about gram-positive, positive begins with P. They will also retain the crystal violet in the gram stain. That is the primary stain. So they will appear purple. And they also have a thicker layer of peptidoglycan. So we're looking at three words that begin with P: positive, peptidoglycan, and purple. So that's how I like to remember our gram-positive organisms. Gram-negative organisms, they will actually retain the counter stain, and they will look red underneath the microscope because that counter stain is safranin or safranin, however it is that you'd like to say it. So as I was saying before, the gram-negative organisms, I think it's kind of like a peptidoglycan sandwich. So here's the inner membrane, and then here is the peptidoglycan, and then here is that outer membrane. So what happens in the staining procedure is that we add the primary stain, then the mordant, which is the iodine, so that we can get this complex to stick to gram-positive cells. But then we also go through a really critical step, which is called the decolorizing step. In that decolorizing step, we use alcohol, and what it does is that it basically wipes away this outer membrane and strips out that crystal violet. So then when we apply the counter stain, then that safranin will actually stick to that peptidoglycan, and it's very convenient that this pen is red right here, and that's why that organism looks red in color. It really looks more pink, but I don't want to confuse you with the P's that we already talked about with our gram-positive organisms. So that's the difference between gram-positive and gram-negative. Acid-fast bacteria, they do have a thin layer of peptidoglycan, but they have this really thick lipid layer, this mycolic acid, and they are really hard to stain because of it. So we actually have to use a special stain, carbonyl fusion, which is a lipid dye that will actually adhere to that mycolic acid. Then we do an acid wash, and this acid-fast bacteria will resist that acid wash and hold on to that pink dye. So they will actually look pink underneath the microscope, like a really bright pink. Now, because of this heavy lipid layer, which is almost like a suit of armor, they are really resistant because of this. So really hard to kill. There's not a lot of disinfectants and antibiotics that will work on them. And because it's tough to get those waste products out and nutrients in, they are very slow growers. And we're going to be talking about the growth curve and generation time. Acid-fast bacteria are much slower growers than our typical bacteria that we will see. So as I mentioned, I promised that we would be looking at a picture. And here is our picture of our gram-positive and gram-negative bacteria. So if you take a look at that top picture, that big purple layer, that's a peptidoglycan. That popsicle stick-looking structure, that's that teichoic acid that has a role in ion transport, also where the bacteriophage can adhere. And then if you look underneath that big purple layer, there's that inner membrane. And so they have a phospholipid bilayer, just like we have a phospholipid bilayer. If you look at the second picture, here's that peptidoglycan sandwich that I was talking about. We've got that inner membrane, a thin layer of peptidoglycan, and then that outer membrane. You'll notice that there's that lipopolysaccharide, that endotoxin, which is part of that outer membrane. So you can see where it kind of looks like that peptidoglycan sandwich. By contrast, if we look at our acid-fast bacteria, there's a thin layer of peptidoglycan, but there's that really thick lipid layer. That's going to act like a suit of armor to help protect them. On the flip side of that, it makes it really difficult to kill them because of that suit of armor. They're also very slow growers. So an example of an acid-fast bacteria that I know that you've heard of is tuberculosis. And tuberculosis affects millions of people worldwide. Sometimes people can actually have tuberculosis and not know because it's such a slow grower that their immune system just kind of keeps it at bay. There are also different strains. The genus of bacteria that do cause tuberculosis are Mycobacterium. We will actually be doing an acid fasting with a Mycobacterium. Don't worry, it is not Mycobacterium tuberculosis. It's Mycobacterium smegmatis, which is not virulent like tuberculosis is. And what happens is that most of the time affects the lungs. And they can actually multiply inside alveolar macrophages. So if you remember the alveoli, those are the functional units of the lungs. And our body can't really handle it. So we end up building like these granulomas, these walls of cells to basically kind of keep it isolated. Most often we think about tuberculosis in the lungs. It can actually spread to other organ systems. This is termed miliary tuberculosis. One of the things that you will hear me talk quite a bit about is that my husband has Crohn's disease. And it took a couple of years to actually diagnose him. And they ended up doing an exploratory surgery trying to figure out what was going on. And when the surgeon came out, he said, if this were another country, I would think that this was tuberculosis. And that was because of the inflammation that was actually in his small intestine. So it can actually affect other parts of the body. Problem with tuberculosis, because it is such a slow grower and it's got that suit of armor, that lipid layer, it's really tough to kill. So there are not a lot of antibiotics that work on it. People that are on antibiotics for tuberculosis are usually on for a long period of time. And of course, we know that some strains are drug resistant. And so that makes it particularly nasty to try to control. So that is an example of an acid-fast bacteria. Another example of an acid-fast bacteria is leprosy, and this is caused by Mycobacterium leprae. Now, I remember when I was a kid hearing stories about leprosies, like people's limbs falling off, and I was like, what on earth is that? It's not quite as bad as that. There are, of course, lesions. I used to hear stories about people being shunned, like they had to live in islands and faraway places because they couldn't be around people. Again, it's not that highly contagious. Of course, people that do have it, they have to be very careful about linens and utensils. But again, because we live in the day of antibiotics, it is not quite the horror story that it was, and we certainly know more about bacteria. Again, just so you're aware, we are not going to be looking at this particular acid-fast bacteria either. And again, just as a reminder, as we are talking about the different types of bacteria, this is more for recognition than anything else. All right, so a little bit more detail on our bacterial structures. So first off, our internal structures. So those spirochetes, those corkscrew-shaped bacteria, in order for them to move, they actually have axial filaments that kind of allow them to twist. So that's how they move. Of course, bacteria are going to have cytoplasm, just like our eukaryotic cell would have cytoplasm. They also need structures to make proteins, and those are going to be the ribosomes. Coming up in a chapter, we're going to be spending a lot of time talking about protein synthesis. And if you've learned protein synthesis and how it works in eukaryotic cells, it's very similar in prokaryotic cells. Of course, because they are bacteria, the enzymes are going to be a little different. The ribosomes are also a little different structure. So just like eukaryotic ribosomes, there's a small and large subunit. Small subunit is 30S, and the large subunit is 50S, and we would term those 70S ribosomes. Now, I know you're looking at 30 plus 50, and you're going, that does not add up to 70. Yes, it doesn't have to do with the math. That S actually means the sediment coefficient. So if you were to spin them down in solution with a centrifuge, that's actually how they would settle out. So this is just to show you that the ribosomes are a little different, and we're going to talk a little bit more about that detail later on. Because there's not a nucleus, they don't have an area that is going to enclose their DNA. Of course, they are going to have DNA, but if we had a very strong microscope, we might see a nuclear region where it was a little darkened. That's going to be their circular chromosome. So their chromosome, they've only got one, is a circular chromosome. Sometimes they could actually have a plasmid. So if I draw out a picture of a bacteria right here, very simple cells. So here would be their circular chromosome, and some of them might have another piece, circular piece of chromosome, which would be a plasmid. And a plasmid is extra DNA. So this is not necessary for the survival of the bacteria. This has nice to have genes. So all of the necessary genes are going to be on the circular chromosome, but some bacteria can have plasmids that can offer some useful genes, like maybe some antibiotic resistant genes. So not all of them have plasmids. Some of them do, and it can offer some useful genes for them. For example, antibiotic resistant genes. Some would have chromatophores. So we would find these in photosynthetic or cyanobacteria. So that means that they would have pigments that absorb light energy so that they could actually make their own food. Because this is more of a clinical based class than an environmental, we're not going to be seeing any bacteria with chromatophores. Again, if we had a really powerful microscope, we might see some small inclusions like little granules or vesicles. And another structure that we might see is an endospore. And I mentioned endospores before. These are not means of reproduction. These are means of protection. And I mentioned that coming up soon, we will be doing the spore stain. And this allows us to see whether or not the bacteria makes a spore. So a bacteria would make a spore when times are tough. So if they think that conditions are rapidly declining, there's not a lot of water, not a lot of nutrients, maybe they've been exposed to a lot of UV radiation or chemicals, they're going to go into this survival mode. So basically, they're going to go into this protective mode where they develop this protein coat, and then they've just got that DNA protected by that protein coat. So they're just dormant, waiting for better times. And a lot of these endospores can actually stay in soil for years and years, like they've even found thousands of years. The metabolically active cell, so the cell just going about its business, doing its job, this is what we call a vegetative cell. They haven't quite gotten the memo yet that they need to go into this protective form. So as I mentioned with the spore stain, that differential stain is going to allow us to see the differences between those vegetative cells, the metabolically active cells, and the endospores, the cells that got the memo, conditions are really deteriorating, and now they need to go into this protective mode where they've developed this protein coat around their DNA. A genus that we see as a spore former is Clostridium. And so we'll give you a couple of examples of Clostridium. If you've ever worked with patients with C. diff, that is a spore former, and that is what makes it very, very tricky to treat and get rid of. So we'll be talking a little bit more about those endospores. Some other external structures. So some bacteria might have pili or pili, however it is that you'd like to say it. These are hollow projections. As we get into our molecular biology chapter, we will talk about how bacteria can actually transfer genetic information. So one bacteria sitting next to another bacteria can actually transfer some genes, like some useful genes, like antibiotic resistant genes. In order to do that, they would need a conjugation pili. And so we'll be talking a little bit more about that process. Some of these pili can actually be attachment pili. So almost like these little stickers that allow them to stick to surfaces. If a bacteria can stick to a cell, that means it's going to be more dangerous to that cell, and that means it can be pathogenic. Some bacteria will have a glycocalyx, a thick layer of polysaccharide on the outside of the cell. A really thick polysaccharide layer, that glycocalyx, would be a capsule. And again, that capsule is going to help to protect the bacteria. And again, if it's protected, it's going to be more dangerous. Some will have a thin layer of glycocalyx, almost like a slime layer, that will allow them to adhere to things. It also helps them to resist drying. We would actually see this with dental caries and dental plaque. So this is what is keeping our dental hygienists in business. Bacteria can also move just like eukaryotic cells do. They can have flagella just like eukaryotic cells can, and a flagella is a tail. Probably about half of bacteria are motile. They can move and they have these flagella. Their arrangement is a little different than eukaryotic flagella. In fact, you can see with the picture how simple they are compared to a complex flagella in a eukaryotic cell. The way that they move is also a little different as well. And we can have a variety of configurations. So Monotrichus would be one flagella, Amphitrachus would be two flagella, one at each end, Lophotrichus, two or more flagella at one or both ends. Peritrichus means that they would be all over the surface. So peri means around. So they would actually have all of these flagella around the perimeter. And they can actually move this flagella in different directions to help them move towards things that are good, like nutrients, or even away from bad things, like maybe chemicals or hazardous things. So this is another picture of the different configurations of flagella. So you can see that Peritrichus where it's got those flagella all around. So here is sort of a generic version of a bacteria where we can see the inside. You can see that chromosome. This one happens to have a plasmid. We can see ribosomes. Again, we're not going to see mitochondria, endoplasmic reticulum. They are not that complex. On the outside, you can see where this one has a glycocalyx. We can also see the cell wall and the cell membrane. And also this one has a flagellum as well. So relatively simple compared to our eukaryotic cell that we will talk about. So I mentioned that bacteria can actually move their flagella in different configurations to allow them to move towards things that are good. Like if there was a plate of chocolate chip cookies on the other side of the room, I would run towards it. Or to steer away from things that are bad. If maybe there's hazardous waste or chemicals in their vicinity. So this movement of either going towards something or away from something is what we call chemotaxis. Now, if you were to Google chemotaxis and look up videos on YouTube, it's really complex. So these bacteria would have receptors. Those chemicals are going to bind to the receptors, and then it kind of kicks off this whole cell signaling to actually move the flagella in different directions. So if the flagella moves, if the bacteria moves their flagella counterclockwise, that would equal a run where it kind of goes towards something. If they were to move them in a clockwise direction, that would result in a tumble. So, of course, you know that I like letter association. So I think of counterclockwise. Counter has an R. Run begins with R. Now, this is not as sophisticated as like running towards something like a plate of cookies. This is what they call more of a random biased walk, where they're kind of like swaying back and forth, almost like, to me, like a toddler. If you've ever seen a toddler learn how to walk, they're kind of like, you know, moving all over the place, kind of like winding around. They can't quite make that beeline yet. So this is not like, okay, they're going to move right towards that nutrient or right away from something that's hazardous. They kind of like go towards something and then kind of tumble away. I did post a video on this just so that you can kind of see the movement of the flagella and how they can do this random biased walk. But it allows them to go towards something or away from something. And we term this chemotaxis. So by comparison, our eukaryotic cell is much more complex. And this is probably what we're more familiar with. So here is a generic eukaryotic cell where it's going to be bigger. And if you look inside, we've got all of these fancy cell organelles. This one has a chloroplast, so this must be a plant cell. We've got the nucleus, where we're going to contain the DNA, mitochondria, where we're going to make ATP. This is much more complex. So how is it that these complex cells came to be? Well, prokaryotic cells were the first on the scene. So they arrived way before eukaryotic cells did. And eukaryotic cells did not evolve until about 1 billion years ago. So this theory of endosymbiosis is how eukaryotic cells evolved from prokaryotic cells. So if we kind of dissect the word endosymbiosis, endo means in. Symbiosis is when organisms are living side by side. And the theory of endosymbiosis is that there were larger prokaryotic cells that gobbled up smaller prokaryotic cells, and they lived side by side. Eventually, those prokaryotic cells living inside that larger prokaryotic cell evolved into organelles. And we can actually see this with our mitochondria and our chloroplast. So if you look at this first set of pictures, we've got this larger prokaryotic cell, and the smaller aerobic bacteria moved into this larger prokaryotic cell. Now, they were both happy because the smaller aerobic bacteria got a home, it got shelter, and it was able to make energy for the larger prokaryotic cell. So they were both very happy living side by side. Over time, that aerobic bacteria ended up evolving into the mitochondria. So it's not like you could actually take out that mitochondria and then it could live by itself. It had evolved to be this specialized organelle. If we look at the evolution of the chloroplast, here we've got that larger prokaryotic cell gobbling up that cyanobacteria. And again, both of them were very happy. Larger cell got some food that cyanobacteria was making for it, and that little cyanobacteria got a home and protection. Over time, that cyanobacteria evolved into the chloroplast. So I know what you're thinking. It's the exact same thing that I was thinking about when I heard this. I was like, well, that's a nice story. But is there evidence of this? Well, there actually is evidence. And this is not a comprehensive list, but this is some evidence for endosymbiosis. So first off, mitochondria and chloroplasts are the same size as prokaryotes. So same size as our bacteria. Okay, that's not really conclusive enough, but wait, there's more. So mitochondria and chloroplasts also have their own circular DNA. Who else has their own circular DNA? We just talked about the DNA in prokaryotic cells. They too have circular DNA. I also mentioned those ribosomes. And I mentioned that math that doesn't quite add up with that large and small subunit. But bacteria have 70S ribosomes compared to the 80S ribosomes of eukaryotic cells. Mitochondria and chloroplasts also have 70S ribosomes. And if they have similar ribosomes, the way that they carry out protein synthesis is going to be very similar. Coming up soon, we'll talk about how bacteria divide. The way they divide is very simple. It's called binary fission, where they double their DNA and then split into two. Mitochondria and chloroplasts divide the same way. I also talked about that gram-negative cell membrane and cell wall, and the mitochondria and chloroplast have that similar double membrane to that gram-negative bacteria. And again, there are some other structural similarities, like the chloroplast is also similar to our cyanobacteria. So there is evidence to suggest that our modern day eukaryotic cells evolved from these prokaryotic cells. So these smaller prokaryotic cells moved inside the larger prokaryotic cells. They were both very happy, and eventually it evolved into the chloroplast and mitochondria. The last thing that I just want to mention is that prokaryotic cells, these cells are going to be prone to the environment and kind of at the mercy of their environment, just like our cells are. And so they need transport. They also have to maintain a high surface area to volume ratio, just like our eukaryotic cells do. So when we talk about cell transport, such as diffusion, osmosis, active transport, we're going to see the same types of things with prokaryotic cells. The one thing that prokaryotic cells excel at is maintaining this high surface area to volume ratio. This is really important so that cells can maintain the exchange with the environment, so that they can get nutrients in and they can get waste products out. So if we have a small prokaryotic cell right here, and it needs to get waste products out, there really isn't a far distance for that waste to travel. And so their surface area, so the surrounding area compared to the volume, is going to remain high, that high surface area to volume ratio. So by comparison, if we were to actually look at a larger cell, like a eukaryotic cell here, much bigger cell, if it's trying to get waste out of the center, it's got a longer way to go. So that means that those waste products can start to build up. That's not a good thing. So prokaryotic cells, they have the market cornered as far as maintaining that high surface area to volume ratio because they're small. In fact, the first rule of maintaining a high surface area to volume ratio is to stay small, like a prokaryotic cell. Our eukaryotic cells are not that small. So how do we do it? Well, there are some other mechanisms to maintain that high surface area to volume ratio. So some cells can kind of elongate and flatten out. So again, if it's trying to get waste products out, now it's got a shorter distance to travel. The other thing is folding things. So this is one thing we talk a lot about in biology. Anytime you fold something, you know that you are increasing that surface area. And then compartmentalization. So if we've got like our mitochondria here and we've got our nucleus, we're extending that surface area. So now we're adding that more surface area by compartmentalizing all of those functions, and that's going to increase that high surface area to volume ratio. Prokaryotic cells, they have that market corner because they can stay small. But again, they are going to be at the mercy of the environment, just like our cells will be as well. So that is a little bit on prokaryotic cells. We are going to be continuing this, of course, looking at our prokaryotic friends in lab as well. And again, I will give you more examples of bacteria, but again, it's more from a recognition standpoint than anything else.